We call it as wood only in Dicot stem, and it is because of the process of formation of vascular cambium between xylem and phloem. Pericycle, as I said, it is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. Cortex is undifferentiated, hence it is called ground tissue. Why it is called undifferentiated? It is not differentiated into cortex, endodermis, etc. Hello everyone, welcome back to session 6 of this chapter called Anatomy of Flowering Plants. Myself, B. Shobharani from the Department of Biology, Vidhyaashram Pre-University College, the Temple of Excellence. What did we study in the last session? Hope you remember, we studied about the anatomy of a dicot root, taking sunflower as an example, and we studied about the TS of a monocot root, taking Zea mays as an example. So we studied the anatomical differences between a dicot root and a monocot root. So what are we going to study in today's session? We are going to study about the stem anatomy. So in the last session we studied about root anatomy. In today's session we are going to study about the stem anatomy. That is TS, transverse section of a dicot stem, taking sunflower as an example, and TS of monocot stem, taking Zea mays or grass also as an example. There's a lot of difference between the anatomical features of a dicot, that is a root, from root anatomy to stem anatomy. Now let us understand in this session how a root anatomy differs from that of the stem anatomy. Moving on to the TS of dicot stem, transverse section of a dicot stem. Here you can see this is the picture called the ground plan, and this is the TS of dicot stem. Example is sunflower, that is Helianthus annuus. Helianthus annuus is the scientific name. So this is the ground plan. Hope you all know that when we build a house, we go for a ground plan. We go for a plan. In a similar way, if you take the ground plan of TS of a dicot stem, it looks like this. So the outermost layer will be epidermis, giving rise to unicellular extensions called epidermal hair, or we can also use the word called trichomes. The next comes hypodermis. The layer below epidermis is called hypodermis. Next comes the cortex. And this is the endodermis. And this is the vascular bundle, or we call it as the stele. Here you can see the arrangement of vascular bundle as conjoined, collateral, that is phloem above and xylem below, and it is open vascular system with cambium present between xylem and phloem. Hope you all remember that we call it as wood only in dicot stem, and it is because of the process of formation of vascular cambium between xylem and phloem. So here you can see this is the cambium, the green color one. This is called as a cambium. Hope you remember what is cambium. Cambium is nothing but a strip of meristematic tissue, or it is otherwise called a few layers of meristematic cells is called as a cambium. Cambium is present in dicot stem but absent in monocot stem. And you can see the arrangement of vascular bundles. They are in the form of discrete ring. They are in the form of a ring structure, and that is called as U-stele condition. It is called U-stele condition. That is, when the vascular bundles are arranged in discrete ring form, it is called as U-stele. And another very important feature is the endodermis. Here you can see the endodermis. Here the endodermis is wavy actually. It is wavy, and this endodermis is called starch sheath. Or why? Because it is called starch sheath? Because it is laden with starch grains. So it is called starch sheath because it is filled with starch grains and it is wavy in nature. So if you take the ground plan of TS of Dicot Stem, taking sunflower as an example, the outermost layer is epidermis, giving rise to unicellular extensions called trichomes. Then just below the epidermis is hypodermis, either made up of, here it is made up of colon-chyma tissue. Then next to hypodermis is the cortex, which is made up of parenchyma tissue. Then next comes endodermis, which is a single layered barrel-shaped cells, which is wavy in nature, made up of starch grains. Hence, the endodermis is also called as starch sheath. Just below the endodermis is the vascular bundle, where we use the word called stele. And the vascular bundles here are conjoined together. Xylem and phloem are together, hence they are called as conjoined, and they are called collateral because phloem is above, xylem is below, and there is cambium, so it is open vascular bundle. And the vascular bundles are arranged in the form of discrete ring, hence you use the word called U-stele. So this is about the ground plan. Now let us go for the transverse section. As I said, once you understand the ground plan, then it will be easy for you all to understand the transverse section. So outermost layer is epidermis. Here you can see epidermis, and it gives rise to unicellular extensions called trichomes. Just below the epidermis is hypodermis. As I said, it is made up of what? Colon-chyma tissue. Next to hypodermis is cortex, which is made up of parenchyma. Next comes endodermis. Here you can see the endodermis. And endodermis here, as I said, it is in the form of wavy structure like this. So this endodermis, which is wavy in nature, is called starch sheath. So here one section of vascular bundle is exposed here in this picture. So in a similar way, you can see the vascular bundles arranged like that. And this is a very important exercise from practical point of view also. So you are given a Dicot stem or particularly the Dicot stem, and you need to take a thin section and study this. So this is how when you take a thin section of a Dicot stem, you get the picture like this. That is the TS of the Dicot stem with outermost layer called epidermis, which gives rise to unicellular extensions called stem hairs or trichomes. Just below the epidermis is hypodermis made up of colon-chyma. Next, just below the hypodermis is cortex, which is made up of parenchyma. Next to cortex is endodermis, which is wavy, which is made up of starch sheath. Hence, it is made up of starch grains. Hence, it is called starch sheath. Next comes the pericycle. Then next comes the vascular bundle. Here we can see in the vascular bundle, phloem is above, xylem is below. And in between there is cambium. So it is what? The arrangement. Here you can see the first form xylem is called protoxylem and the later form xylem is called metaxylem. But this is called as pith or it is called medulla. Pith or medulla. Here you can see the protoxylem is towards the pith and metaxylem is towards the endodermis. So it is called endarch. It is called endarch because protoxylem is towards the pith, metaxylem is towards the endodermis. For example, if it is vice versa, protoxylem is towards the endodermis and metaxylem is below, it will be called as what? Exarch condition. Here it is endarch condition. So what are the very important parts of TS of Dicot stem? Epidermis, giving rise to unicellular extensions called trichomes. Just below the epidermis is hypodermis, made up of colon-chyma. Next comes cortex, which is made up of parenchyma. Next comes endodermis, which is a starch sheath, or it is made up of starch grains, hence the name starch sheath. Next comes pericycle, which is made up of again parenchyma. Next comes, this is what we call it as stele. Stele region comprises of what? Vascular bundles. Comprises of vascular bundles, namely xylem and phloem. Conjoined, collateral, open. Why we call it as conjoined? Because here you can see xylem and phloem are together. So you can see this cambium, phloem, xylem. So this is called conjoined, collateral, then open vascular bundle because cambium is present. And pith and it is called endarch. it is called end arch. Why it is called end arch? Because protoxylem. What is protoxylem? The first formed xylem, which is smaller in size, is called as protoxylem, and metaxylem, the later formed xylem, is called metaxylem, which is bigger in size. Here you can see the shape. So it is big. Here it is small. So this is about the anatomy of the TS of dicot stem. Outermost layer is what? Epidermis. Epidermis is covered with thin layer of cuticle and has trichomes and few stomata. So as I said, even stem also is also involved in what? Transpiration. The cells arranged in multiple layers in between epidermis and pericycle constitute the cortex. Cortex has 3 subzones. What are the subzones? Hypodermis, cortex, endodermis. Cortex is made up of what? Parenchyma. Here it is, hypodermis is made up of what? Collenchyma. Endodermis, it is innermost layer of cortex. Cells are rich in starch grains and are referred to as starch sheath. Here you can see. So what is the TS of dicot stem? First one is epidermis. Second one is hypodermis. Epidermis will give rise to trichomes, or we use the word called stem hairs. Hypodermis. Next comes cortex. Hypodermis is made up of collenchyma. Cortex is made up of parenchyma. Next comes endodermis, which is a starch sheath. Next to endodermis is pericycle. Next comes the vascular bundles. Here the vascular bundles are conjoined, collateral, then open vascular bundle. So this is how it looks. So epidermis, hypodermis, cortex, endodermis, pericycle, stele, that is the vascular bundle. We can also use the word called stele. Next comes 7th one is what? Pith or medulla. So this comprises of stele. Then this is pith. So this is about the TS. So we'll see. Pericycle, as I said, it is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. Medullary rays. So here this is called as the medullary rays. Medullary rays. Also we can use the word called pith rays. Right. So medullary rays, a few layers of radially placed parenchymatous cells present in between vascular bundle. A large number of vascular tissues arranged in a ring, hence it is called U-stele. Why it is called as U-stele? Because it is arranged in the form of a ring here. So it is called U-stele because the vascular bundles are arranged in the form of ring-like structure, hence it is called U-stele. The central portion of the stem constitutes pith. So this is the pith region. Here this is the pith. This is the pith region. So pericycle, it is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. Here you can see the pericycle. Medullary rays, as I said, the medullary rays, a few layers of radially placed parenchymatous cells present in between vascular bundles. A large number of vascular tissues arranged in ring form, hence it is called U-stele. In dicot stem it is U-stele, but in monocot stem it is atactostele because it is scattered. Then each vascular bundle is what? As I said, conjoined together, then open and end arch protoxylem. Why it is called end arch? As I said, it is end arch because protoxylem is towards the pith and metaxylem is towards the endodermis. It is called end arch. So the central portion of stem constitutes the pith. So this is a very important concept from examination point of view, both from theory point of view as well as from practical point of view. Usually this is asked in the practical examination. It is a guarantee question in the practical examination. So also in theory also. So this is all about the TS of dicot stem. Let us move on to study how the monocot stem, TS of monocot stem differs from that of a dicot stem. TS of monocot stem, what is the example we are going to take? It is Zea mays, or even we can take it as grass, right? So this is how it looks. If you take the, here you can see this is called the ground plan. This is the ground plan, and this is the TS of monocot stem. How does it look? See how different it is from that of the dicot stem. I think we can just go through this. See how it looks. So this is totally different from that of a monocot stem. So here we can see the outermost layer is epidermis. Next comes hypodermis. Next comes ground tissue. As such, there is no cortex, there is no stele. So the total tissue that is found, we call it as ground tissue, or also we can use the word called fundamental tissue. Fundamental tissue. And the vascular bundles are scattered in the ground tissue. So here you can see the vascular bundles are scattered in the ground tissue. And one more thing you can observe, these are the vascular bundles. The vascular bundles towards the periphery are more in number and smaller in size. Smaller in size. Don't you observe? Here if you see that the vascular bundles that are found towards the periphery, that is towards the margin, are smaller in size, more in number. And those towards the center, here you can see they are bigger in size and less in number. Less in number. Here see, so this is a bigger one and this is a smaller one. So here you can see the vascular bundles are just scattered. So it is called atactostele. Here you can see atactostele. In dicot stem, it is what? It is eustele. That is, the vascular bundles are arranged in the discrete form of ring. Here you can see they are arranged in the form of discrete ring. Here in monocot stem, they are scattered. They are scattered. Hence, it is called as atactostele. So now let us see the TS. Here the outermost layer is cuticle, epidermis, hypodermis. Hypodermis, here it is made up of chlorenchyma, that is the modified parenchyma. Then next comes the vascular bundles. Here you can see this is the ground tissue. The vascular bundles are just scattered in the ground tissue. And here you can see the protophlorium and the metaphlorium here. And this is protophlorium, this is metaphlorium, this is metaxylem, protoxylem, and protoxylem lacuna. Here the protoxylem is crushed to form a cavity called, it is called lysogenous cavity. It is called lysogenous cavity. And here you can see the bundle sheath that is made up of sclerenchyma. So this is how the transverse section looks. What is the anatomy of TS of dicot stem? Outermost layer is epidermis. Epidermis is covered by cuticle. Hypodermis is made up of sclerenchyma. Here it is sclerenchyma or sometimes even chlorenchyma. Then cortex is undifferentiated, hence it is called ground tissue. Why it is called undifferentiated? It is not differentiated into cortex, endodermis, etc. So stele. So it is called undifferentiated cortex. Why it is called undifferentiated? Because it is not differentiated into cortex, endodermis, pericycle, stele. It is just a ground tissue. Right. Next, the vascular bundles are scattered here. Here you can see they are scattered in the ground tissue. Hence it is called atactostele. Moving on to the further concepts of this monocot stem, each vascular bundle is covered by a bundle sheath cells. Here you can see bundle sheath cells which is made up of sclerenchyma mainly to give mechanical support. So each vascular bundle is covered by bundle sheath cells. Next, vascular bundles here are conjoined and closed. No cambium. Here the vascular bundles are conjoined and closed. And one more thing, the protoxylem is crushed to form a lysogenous cavity. Lysogenous cavity. So, see here, water containing cavities is called lysogenous cavity. And this protoxylem which is crushed to form a lysogenous cavity is also called as water cavity. It is also called as water cavity, which you don't find in dicot stem. So this is very, very unique, only present in monocot stem. And there is no cambium. So you don't find secondary growth in monocot stem. Secondary growth is absent in monocot stem, but present only in dicot stem. So peripheral vascular bundles, as I said, the vascular bundles which are towards the periphery are smaller in size and more in number. And those towards the center are bigger in size and less in number. Bigger in size and less in number. Hope you have understood the anatomy of a dicot stem and a monocot stem. So this is a very, very important concept from examination point of view and also from practical point of view, as you are going to do this in the practice. You are going to take a thin section of a dicot stem and a monocot stem, and you are going to learn the internal structures or the anatomical features, how a dicot stem differs from that of a monocot stem. So I'll be back in the coming session with some more interesting concepts of, I think we have come across root anatomy, then stem anatomy. In the next session, let us study about the leaf anatomy. How does a dicot leaf differ from that of a monocot leaf? So I'll be back with some more concepts in the next session. Till then, thank you.